So, good evening, everybody, and welcome back. So I haven't been doing this job for very long, and already I love it. Because tonight I have to do one of the coolest things I've ever got the opportunity to do in my career, which is to give the Waddington Medal to a legend in developmental biology. This person, their ideas and their influence is kind of woven into the fabric of the discipline, and there is nobody in this room who has not been influenced or touched by his contributions in some way or other. So he's made many contributions, as I've said. I've put together a few slides which touch on some of those contributions in different spheres, important spheres of developmental biology. So some of the key questions that he's answered. What is the most important life event? He's very famous for having sorted out this question. Could it be birth? Notice his hat, it'll recur later. Death or marriage, or maybe it's gastrulation. Well, he's answered this question very impressively. Another important question, if you have a knockout mouse, what kind of phenotypic assay would be an appropriate assay to try to discern its phenotype? Might you be interested in its activity cycles or its food preference, or maybe you should take it to the opera to see what its opera preferences are. And he's very eloquently pointed out that actually all these and a whole load of more things that you haven't thought about are necessary before you can declare your knockout mouse to have no phenotype. And he is particularly enthusiastic about the opera assay for reasons of high culture, no doubt. Then the thing that perhaps we all more immediately associate with his name is the wonderful French flag hypothesis, which has been a key way of thinking about the interpretation of positional information, and particularly gradients in biology, which is still discussed today in many, many developmental contexts. And then beyond that, of course, he's made very many important contributions in terms of training. We've all read his textbook, and another important question he's answered is how many famous developmental biologists can fit on a roof terrace. This is a picture of his lab, kindly provided for me by Cheryl, who is here, showing his lab clustered onto a roof terrace in one of their many group meetings, and you can see the extraordinary contribution he's made in terms of the people he's trained, because just in this group you can spot many very well-known developmental biologists, including many previous winners of the Waddington Medal. So I don't know, maybe there's a competition afterwards for who's managed to jot down the names of all these people. And then another extraordinarily important question that he's answered, which I will end on before welcoming onto the stage. When is the best time of year to give a party? So here you can see the recurrence of the Father Christmas hat. And clearly Christmas is the best time of year to have a party. Although if you're a developmental biologist, as I mentioned earlier, pretty much any time of year is a good time of year to have a party. So I'm sure you've all worked out who we're talking about by now. And it is my tremendous privilege and honour to present to Lewis Wolpert, who is not only the Father Christmas of developmental biology, but also the father of developmental biology, this Waddington Medal. Lewis, welcome.
We'll come back. First of all, thank you very much. I'm very flattered and honoured to get the Waddington Prize. And thank you all for being here and for the applause. And Ottoline, thank you so much. I even remembered your name for 2 minutes. Thank you very much indeed. Now, I'm going to talk a little bit about, I spent my life in developmental biology. How did I get there? I didn't get there in a very straight sort of way. I'm a South African. And I went to university in Johannesburg and studied civil engineering, which I wasn't mad about, but it wasn't too bad. And then I became the personal assistant of the director of a building research institute for 2 years and studied soil mechanics, which I wasn't mad about either. I then left South Africa. I was too involved in politics and I was frightened. I knew Nelson Mandela and sold his communist newspapers every Sunday and so forth and so on. And I wanted to get away from the politics, and I also wanted to get away from my parents. So I hitchhiked up Africa. It was quite exciting for 4 months with a friend of mine, and ended up, we even took an Arab dhow from Mombasa to Mukalla, and eventually I ended up in Israel doing more soil mechanics. Wasn't very keen on that. And eventually I came to London, and what did I do? Soil mechanics at Imperial College. And I wasn't at all pleased about that. I really didn't want to be a soil mechanic. I wanted to get into something biological. I thought very seriously about doing medicine, but I was too old at that stage, and so I didn't know what to do. But a friend of mine in South Africa, Wolpert knew that I had this problem. And he read in the paper about 2 well-known scientists who was working on the mechanics of cell division. And Wolpert wrote to me and said, "Lewis, this is what you must do. You must take your mechanics and work on the mechanics of cell division." And I went to King's College, to the professor where he was going, Jim Danieli, and said I wanted to do this. And he said, "Yes, okay, you can do a PhD on this." And I worked very hard. I had to pass exams in zoology, I had to learn zoology, and I had to start working on the mechanics of cell division, working on sea urchin eggs. See, that's a sea urchin egg. And I can, or the earlier, that's a sea urchin egg too. That's what I worked on for studying cell division when it divided. And I was doing quite well. I used to go to Sweden to work there, and I liked working on it really quite a lot. And I did eventually get my PhD. The model that I came up with was wrong, I have to tell you. But it wasn't bad. And in fact, I just wrote a little paper recently, about a year ago, putting a little bit of my model in to the current model. I won't bore you with the mechanism. And so there I was with a PhD in cell biology. But while I'd been working on sea urchin eggs, I'd also been... that's Hans. This is an experiment of Hans Drisch, in which I think it's very important to recognize that if you take the 2-cell stage and separate them, this one will give rise to a larva which is half the size of that. And I thought that was absolutely remarkable. And I began to think about that very thing. This is an experiment that Drisch did over 100 years ago. And Drisch's view about this, he thought that there was a special, magical, impossible system called entelechy, whereby the cells knew their position and therefore knew what to do. Total nonsense, but nevertheless it was interesting. And so I began to think about things. I thought I'd like to understand this and really work on it. And I began, for example, to think I couldn't go to the marine station every year as I had been going because I got married. That's one of the things with marriage, and I had 4 children under 5. I won't talk to you about that. And a lesbian wife. Complicated life. Complicated life. That's all. The children are fine. The children are fine. My behavior wasn't that good. Don't let's get involved with that. And I decided to try thinking about organization. And we did some experiments on Hydra. There was this positional gradient here, and this is the inhibitor which prevented the head from setting up. I'm not going to take you through it. It wasn't great, but at least I was beginning to work on those things. But as I thought more and more and more about Drish's experiment, and I really thought very hard and had long discussions with my students, we came up with the French flag model. And because the real point about a flag is no matter what its size is, its pattern is identical. And so I thought this is somehow what embryos do. And so here is the French flag model. Here's the group of cells, and here you are. You divide them up. And if you make it bigger or smaller, so long as the cells know their position, and that's where positional information comes from, everything will be all right. And so that's what I spent a lot of time working on. And I decided now not to work on Hydra because I'd moved to a medical school. In I don't know what year I moved, I became a professor at the Middlesex Hospital Medical School. I think when I was 36 or something, I don't know what I was. Anyhow, I went there, and it was a medical school. I couldn't work on Hydra there. So I decided to work on the limb, on the development of the chick wing. And that's what I decided to work on. But also there was other evidence for positional information. Early published, not my work at all, set of experiments on the cockroach leg. Just have a look. Here's the leg, and you number it, and you think there's a gradient that gives that. And if you cut it here, like that, it regenerates all right. But look what happens when you make a cut where you join the 4 to the 2, it makes it even longer. And I think this is some of the best evidence that I know that cells really do have positional values. I'll come back to regeneration a little later. But I think this is a remarkable set of experiments and really are absolutely excellent. And you can see that when you put the 1 next to the 5, 2 and 3 and 4 intercalate. So I was very taken. I was very taken with those experiments. And then there's a nice experiment, and once again not mine, of the following kind. That you take a piece from the early French flag and put it in a region of the developing French flag. And what does it form? It doesn't form this region, it corresponds to this region, because it corresponds to that region there. And there is an experiment, a classical and important experiment, which supports that very strongly. That's an experiment of the following kind. As you know, there's the gene antennapedia, which turns the antenna of the fly into a leg. Okay? And so what the experiment was to actually have a look more closely at that. And if you just think what happens is just to take a small region of the antenna and convert it into leg, it made the right piece of leg corresponding to its position along the axis. And that meant that positional information in the antenna and the leg were identical. They just interpreted them differently depending on what genes there were there. So it once again was a very nice example of positional information. It would have pleased me anyhow. And then, as I said, I decided that I was going to work on the chick wing bud because, at least for medical students, that seems a little bit better. I spent most of my life working in medical schools, and I have I don't know how many doctors there are in the audience. I'll tell you my joke about having worked with them for 30 or 40 years. I want to see doctors when I'm ill, and I want to see doctors when I'm at a party. I never want to see them on a committee. Maybe you haven't had that experience yet. So here we have the wing bud and the leg bud, and at the tip of each of them there's an apical ectodermal ridge that secretes a signal. And here we go. And this is what it looks like. Have a look here. So here is the apical ridge here, and underneath it our model says there's a proliferating region here, which we call the progress zone. And there's also discovered, not by us, but by other people, a region at the posterior margin of the limb, which is the polarizing region. That's the proximal region, so there we are. And then this limb develops and cartilage begins to develop, and you get the various cartilage regions in the limb. And our model was, in terms of positional information, is that that polarizing region set up a gradient. And it turned out that that gradient was sonic hedgehog. And this was the normal gradient. And at this level, you make a digit 4, at this one a digit 3, and at this one a digit 2. So that when you put in another polarizing region, you make a mirror image gradient. And if you put in a very small concentration, you just make an extra digit 2. Now, this looks plausible, but the evidence that it's really a diffusible gradient, there's just not good evidence for this. We really don't understand what's going on there. First of all, there's now evidence that sonic hedgehog doesn't diffuse. It's actually carried. It's not our work at all. It's carried on long extensions of the cells there to different regions. And we just really don't know how reliable this model of the gradients are. It fits things very nicely, but we don't really know the details of what's really happening there. And then what happened was, I was working on this, and we were invited by Waddington, who started having a set of meetings on theoretical biology at Cerbalone, which is on the, which sea is it on again? I never remember, never the matter, on the coast somewhere. And we went along there, and I told them my ideas, and they went down quite well. The ideas went down quite well. And then later that year, or the next year, I went, and I became very friendly with Waddington. Waddington became a friend of mine, and I liked him very much indeed. He was very helpful. I used to stay in his flat in Edinburgh. We got on very well. But I went to Woods Hole, where I was working on my sea urchins. I don't think I showed you a picture of sea urchins, did I? Did I? No, I didn't. They disappeared. No matter. I went along to Woods Hole and gave my talk at one of the major Friday night lectures. No one would talk to me that evening or the next day. You may say I'm making this up. No one would speak to me that night or the next morning. They hated my model. They didn't tell me why, but the thing. And next morning, as I was bathing, you'll never believe who I met next to me, Sidney Brenner. If you don't know who Sidney Brenner is, then you don't know the only genius I know. I knew Sidney from South Africa because he was at the university with me and a few years older. And Sidney found me crying in the water and told me to pull myself together, that he and Francis Crick liked my ideas, and I was not to worry about what these people thought or not. It saved me completely. Instead of going into severe depression, I was absolutely saved by Sidney. It made an enormous difference to my life. And so that was very important for me. And then there were other experiments we did. And for example, we did this experiment which other people had done, in which you take the early limb bud, take off the ectoderm, separate all the cells, just shove them back in a lump, put them back, and they actually make quite nice digits, quite nice skeleton. How on earth could that be? And the only explanation can come from Turing's model. Are you all familiar with the Turing model? You're not all nodding. I will tell it to you. You know, he's the one, he's very famous, he's just in this recent film, but he also made an enormous contribution. And here's the Turing model. You've got two key molecules, an activator, which activates itself, but it also activates an inhibitor, which inhibits the activator. And if you choose the right parameters, it makes peaks. And if you make it the right thing, it will lead to a series of peaks, which can give rise to the digits. And very recently, the Turing network has been implemented by BMP, SOX9 and WINT and drives digit specification. This has been done in a recent paper in Science by these 4 people. And that's very encouraging. And don't just look at this here. This is what it looks like in the normal limb. It's more complicated there. So that's the Turing model, and it's really highly, highly imaginative. And then we had a very different model for the proximo-distal axis. That's based on the progress zone. So remember, I told you the apical ectodermal ridge here specifies this proliferating region, the progress zone. And the cells measure, it's about time. It's how long the cells remain in that zone that determines. I'm going to sit down before I get tired, children, so I'm going to go and sit down over here. Sorry about that, that's all right. Well, that's great, kid. I need to be able to see what you don't mind, no. So, you can hear me still, can't you? Yes, good. So, depends how long the cells are in the zone that determines their position. Cells that have only been in a short time will be very proximal. Cells that have been in a long time will be very distal. You can see it there. Okay, so, but you get the general idea of the progress zone. It's how long the cells remain. The cells are all dividing. They measure how long they've been in when they leave. And the longer they're in, the more distal they are. So that was our model. I just want to tell you that these models that I'm telling you about were not wildly accepted. I mean, a friend of mine, Cliff Tabin, hated them and has written many papers attacking them. But I think he's come around to, I think I have the illusion that he's more sympathetic towards them now. Do you think that's true? I don't know. But it hasn't been an easy time. It's been quite a long battle with these ideas. But one of the, I think, the nice experiments that I did, by the way, I never do an experiment. I am a theoretician. I hate doing experiments. My skill is to get other people to do the experiments. Okay, I mean, but I work very hard on the results and I work very hard in preparing it. But I don't like manipulating things with my fingers and hands. I did it for my PhD, of course, when I did the mechanics of cell division, but it's not something that I'm good at. And I had one technician, Marta Hornbrook, for about 30 years. Marta and I, she worked together. And I was still very friendly with Marta. And her name was on all the publications. I never published anything that she did without putting her name on the paper. And we got on very well. And also, I had wonderful PhD students and postdocs. I had Cheryl Tickle, by the way, who is a Waddington Medal winner. I also had Jim Smith as a PhD student. I had Neil Vargason here as a PhD student. I was very fortunate at the Middlesex to have excellent students. And my skill was to be able to persuade them to do the experiments, and then I would do the analysis and help write up the paper. Okay, so here's the experiment that we did. I'm glad you understand. So here it is. Here's the progress zone with 25 cells. And at the first cell division, 25 cells come out, and they're going to be slightly more proximal. And the next cell division, another 25 cells will come out, and they'll be slightly more distal. But then you do an experiment, and that was my plan, is that you kill cells in the progress zone. So there are only 5 living cells in there. So at the first cell cycle, only 5 cells come out. You're not going to get much humerus or radius and ulna in there. The next cell cycle, 10 come out. No, 7 good ones come out. It's getting a little better, but not much better. But you do see that the progress zone is becoming repopulated. And after another few cell divisions, it will be back to normal. And this means that when you do this experiment, you will lose proximal structures, but the distal ones will still be there. And that's exactly what we found, I'm very pleased to say. And the next slide sort of shows one. Here's an example. Where is it? This one here. you see, when you did the experiment, then we grafted onto another limb, you only got distal structures. And of course, and Neil Vargason is something that he works on, we thought that this is probably the way thalidomide acts. That thalidomide interferes with the vascular system so that cells in the progress zone die, and then you get this result. You get a child with just a hand sticking out of the shoulder. But it's Neil Vargason's subject, and you should really ask him about it. And then the other thing, of course, we obviously did, is we thought quite a lot about was how muscles developed. And it's quite surprising how muscles develop, because muscles don't have positional information. The muscles in your arm move in from the somites, and it doesn't matter which somite they move in from. They'll give you the perfectly reasonable muscles in your arm. So what happens is they move in and they'll form a mass up here, and then they'll migrate in this region here. And of course, because all those cells have positional values, they tell where the muscle cells must stop. So the pattern of the muscles is actually in those cells in there. And the cells migrate until they're given instructions not to move. So that's once again, the cells have all positional information in the limb. The muscle cells don't at all, and they just go where they do. And incidentally, the way muscles join up with tendons and things like that, they don't have any positional identity. They just join up with what's very close to them. They don't really care. They're very democratic. They have no positional values whatsoever. And then it's just worth also having a look at growth. Because if you look at the region in the limb, the early limb bud, which grows, it's really quite a small region. And here's the humerus region. It starts off very small and grows like mad. And the ulna does the same, but the wrist hardly grows at all. But initially, these regions are of an identical side, and I can only assume it's positional information, it's their positional value which determines how much they're going to grow. So that's really another example of positional information. And then an important point is this. Our limbs grow 100 times bigger over 16 years, but they have the same length, but there is no communication. Could you all do me a favor, please? Stretch out your arms. How similar are they? There may be one or two of you that it's not the same. But just think that those limbs have grown for 14, 15 years since they were in the embryo, and they have almost identical length. And no one has a clue as to why that should be so. I published a paper on it. Nobody, not a single person, has come up with an explanation. Not one single person has come up with one. And my own interpretation is only because a lot of cells are involved in the growth of the limb, that they're simply so reliable. There's no evidence for communication between the limbs, so that they do have the same length. The only explanation I can come up with is that the cell's behavior is so reliable, and there are a lot of cells in the development of the limb, and that's why they simply behave in exactly the same way. But you may have a different model altogether, and I'd be delighted to hear it. And while we're talking about models, let me tell you the other model that I'm desperate about is what is the basis of positional information. I'll come back to that in a moment. We just, if you just go back to Antennapedia, the leg and the antenna, we know that positional information is the same thing. But what is the molecules that actually establish those positional values? And there are a lot of models around which try to explain how you can reliably set up gradients. Let me say that the thing I hate most is diffusion. Anyone who has a model in which the gradient is based on diffusion, I simply don't believe. Diffusion is far too unreliable for setting up a reliable gradient. It's more related to cell contact and some sort of Peter Lawrence's models where the cells actually talk to each other directly, and that sets up the gradient. But the blunt fact is, after all these years and all this work, we do not know, except for one exception, which I'm going to talk about in a moment, one exception, we do not know the molecular basis of positional information. And it's worth looking here at what happens in regeneration. So here's an amphibian limb, and you cut off the limb, and it regenerates. So there's the limb, and you cut it, and it regenerates. What's going on there? Well, the answer is, it's positional information. So here's a blastema at this level here, and here's a blastema more distally. If you take this blastema and put it on there, it will intercalate all the things there. And that's another nice example of what's going on with positional information. We don't know the molecular basis there, but it's quite good. But Jeremy Brock's has made a very important discovery, a set of discoveries, and that is that he found that a membrane protein called PROD1 has a 5-fold difference along the nucleolum. This is equivalent to about a 2% difference over each cell. So that's the only example we know where we have a molecule that is graded and can provide positional information. I'll show you the evidence that it does provide positional information. It's not my experiments, it's Jeremy's. So just have a look. Let me tell you, retinoic acid increases the concentration of PROD1, okay? So if the limb is cut here, yes, normally it would regenerate there, but it's treated with retinoic acid, so its positional value is equivalent to here, and so it makes a whole new limb. Are you with me there? I hope you're understanding what I'm talking about. So retinoic acid raises positional thing of PROD1, and therefore when you cut at the tip and you treat with retinoic acid, PROD1 goes up, and therefore it thinks it's the proximal region and therefore makes a whole limb, which is really quite strange. So those are the main things that I've done in developmental biology. There have been a few other where I have had various models which many people don't take very seriously. I have a model for the origin of multicellularity. I'm not going to tell it to you. I have what is my other model? Oh, I have a model for left-right asymmetry based on an F-shaped molecule. Metamorphosis. Yes, I have quite an interesting model for metamorphosis too. I don't think anyone takes that seriously either. I've also written books, some of which nobody takes seriously either. I think I have quite an important book on the origin of religion. Nobody takes it seriously at all. I've just written a book on the biological differences between men and women. It's hardly had any reviews whatsoever. The one book that I have written, which has been well received, was Malignant Sadness. I had a very severe depression about 15 years ago, and I didn't have a clue what depression was about, but I was in hospital for 3 weeks at the Royal Free, and when I came out, my wife at the time hadn't told anyone that I was depressed, because she thought the stigma was too great. And so I thought this was ridiculous, and I decided to find out what depression was about, but I knew nothing about depression. And I wrote my book, Malignant Sadness, and it does really argue, and once again it's certainly not accepted, that depression is really sadness become malignant. It hasn't got any advantages. You don't want to lecture on depression now. I've also written a textbook called Principles of Development. It's just come out. The fifth edition has just come out. I didn't write a single word. It was all done by my colleague Cheryl Tickle. I have somewhat dropped out of, I'm sorry, of developmental biology. It's partly old age, but it's partly that I find when I look at the journals, I find them so unbelievably boring. It's all detail. And I'm not interested in detail, I'm interested in general principles. And so I don't take the journals anymore. I glance at them occasionally on the web, and I occasionally look at stuff that relates to my ideas. But I do find it really very disappointing, and I don't think. And so I've really dropped a bit out of developmental biology and have focused more on my writing. And I have written a general book on developmental biology or cell biology for the public as a whole, and that hasn't done too badly. I also wrote a book on old age entitled "You're Looking Very Well." That's what we say. And I think those are the main things in my life. And I was interviewed today, and the lady said to me, what's the thing that you think about most? And I say it's my memory. My memory is too terrible. And I'll tell you, I don't recognize people. Old friends I don't recognize, and I can't remember their names. And my favorite story when that happens, to make them feel better, is I was staying in my son's house in Cambridge, alone. And I went down for breakfast one morning, and I found a young woman there doing some washing up. And I said, "Hello, and where are you from?" She was my granddaughter. I didn't recognize her. That's the way things go. Thank you very much for listening to me. All right.